So picture a gymnast stepping onto the floor for their routine. They take this deep breath, they sprint down the mat, and they just launch into this massive double backflip.
Oh, yeah. Pure explosive power.
Exactly. They're basically defying gravity with raw, fast twitch energy. But then, like, mere seconds later, they land and immediately drop into a perfectly flat fluid split right on the floor.
Right, which is just wild to think about.
It is. And I mean, we watch this happen during the Olympics, and we don't really think twice about it, right? We just assume, well, that's what elite athletes do.
Yeah.
But biologically speaking,
It makes no sense.
right, that combination of extreme structural flexibility and explosive high twitch power is a massive physiological paradox.
Yeah.
Because those two physical traits are essentially mortal enemies.
They really are. I mean, it is one of the most frustrating fundamental conflicts in all of human biomechanics.
Yep.
Because when you optimize the human body for one of those traits, you are almost always actively sabotaging the other one.
So it's a zero-sum game, basically.
Pretty much. If you want maximum power, your muscle and tendon structures need a certain amount of stiffness. They need to act like stiff springs. But if you want maximum range of motion, you need compliance. You need looseness.
And that biological conflict is exactly what we are getting into today. So welcome to this deep dive.
Right.
Because today, we are looking at a brand new piece of research that might have actually found a biological loophole to this entire system.
A really exciting one, too.
Yeah, it's fascinating. We are analyzing a 2024 study titled The Effects of Static Stretching 2 hours prior to a traditional warm-up on performance.
Right.
And this is authored by Ashley Shea, Mohammadmahdi Bahrami, Mahta Sardroodian, and David G. Behm. Published in the Journal of Sports Science and Medicine on September 1st, 2024.
It's a really critical piece of literature to examine, honestly, because stretching, despite being something literally all of us do, it's one of the most misunderstood areas of sports science.
Oh, completely.
Yeah. For decades, the advice has just shifted back and forth. And it leaves athletes, coaches, and, just everyday gym goers completely confused about how to prepare their bodies.
So true.
But this specific study sets out with a really accessible but highly rigorous academic approach. And it answers a question that applies to you, whether you're a competitive athlete or just someone trying to optimize your physical performance and stay injury-free.
Okay, let's unpack this. Our mission today is to discover if there is a scientific loophole. Right. Like a specific, perfectly timed protocol that allows you to get that maximum deep flexibility without sacrificing your explosive strength and power.
Exactly.
But before we get to the actual loophole they discovered, we have to talk about why static stretching became such a villain in the sports science world in the first place.
Yeah, the backstory is important here.
Right. Because for a long time, the golden rule of fitness was to do a lot of static stretching right before you work out. You reach down, touch your toes, hold it for a few minutes, and then go run your sprint or lift your weights.
That was the classic advice, yeah. Everyone did it. But the academic literature over the last 2 decades has definitively proven that this is a terrible idea.
Terrible idea.
Oh, absolutely. Especially if you are about to do anything requiring power or speed. The consensus in the literature is super clear. If you perform prolonged static stretching, you are going to impair your performance.
And how do researchers define prolonged in this context?
Basically, holding a single stretch to the point of discomfort for more than 60 seconds per muscle group.
Wow, just 60 seconds.
Yeah, that's the threshold. If you
Ugh.
pass that, you will absolutely increase your range of motion. You will get more flexible in that moment, but you will actively impair your physical performance immediately afterward.
So let's put some numbers to that impairment, because I don't think people realize how significant the drop-off really is. What are the actual metrics that take a hit when you hold a stretch for too long before an event?
Well, we see significant reductions in what we call MVIC.
Okay, MVIC, what is that?
It stands for maximal voluntary isometric contraction. That is basically your absolute peak force output.
Gotcha.
So if you're trying to push against an immovable object with everything you have, your MVIC drops after prolonged static stretching.
Wow.
But the impairment goes beyond just static force, too. It immediately reduces your vertical jump heights, specifically your counter movement jumps and drop jumps, and it actively slows down your sprint speeds.
See, that is the part that completely scrambles my brain.
Yeah.
You are doing something that is universally known as warming up your muscles, but the physiological reality is that you're making them fundamentally weaker and slower.
It's totally counterintuitive.
Yeah, because if I am an athlete, my goal is to prime my body, right? Not sedated.
Mhm.
Why on earth does the human body react to a deep stretch by shutting down its own power output?
Well, the underlying mechanism is where human physiology gets incredibly interesting. When you hold a deep, uncomfortable stretch for a long time, you are triggering this whole cascade of neurological safety mechanisms.
Okay, safety mechanisms. Like
Yeah.
what's...
The primary one is called the disfacilitation of afferent muscle spindle reflex activity.
Oh, wow. Okay, I'm going to try to reason through that one because disfacilitation of afferent muscle spindle reflex activity is a mouthful.
It is, yeah. Go for it.
Okay, so if a muscle spindle is essentially a microscopic sensor inside the muscle belly, right?
Right.
And it detects how fast and how far the muscle is stretching. So when those sensors sense danger, they trigger a reflex to forcefully contract the muscle to protect it from tearing. Like when a doctor taps your knee with a hammer and your leg kicks.
Exactly. That's the stretch reflex.
So if you are holding a deep stretch for 3 minutes, are you basically numbing those sensors?
That is an excellent way to conceptualize it, actually. You are continuously bombarding those microscopic sensors with a stretch signal until they essentially become desensitized.
Okay.
You're basically dulling the nervous system's ability to fire that muscle rapidly. But the neurological dampening doesn't stop at the muscle itself.
Wait, it goes further.
Oh, yeah. Prolonged static stretching also suppresses what neurologists call persistent inward currents, or PICs, up in the spinal cord.
Okay, let's explore these persistent inward currents.
Yeah.
I imagine the signal starting in the brain, traveling down the spinal cord, and then hitting the muscle. Where do the PICs fit into that chain of command?
So PICs act as biological amplifiers, and they are located right in the neurons of your spinal cord.
Biological amplifiers, I like that.
Yeah. So when your brain sends a signal saying jump, that signal might start out relatively small. The PICs in the spinal cord amplify that electrical signal so that by the time it reaches your leg muscles, it is a massive, booming command to contract with maximum force.
Okay, that makes sense.
But when you hold a deep, prolonged stretch, the feedback loop from the muscle actively suppresses those amplifiers in the spinal cord.
It sounds like you are literally turning down the volume knob on your brain's ability to communicate with your muscles.
That's exactly what you're doing.
It makes me think of taking a thick, powerful rubber band and stretching it as far as it will go and just holding it there for 5 minutes. You are breaking down the structural integrity.
Right.
When you finally let go and try to snap it, it doesn't have that violent, explosive recoil anymore. It just feels loose and floppy.
That rubber band analogy beautifully illustrates the dual problem here.
Yeah.
You've increased the structural compliance of the tissue, which is the flexibility, and that's a good thing.
Right.
But you have simultaneously killed the elastic energy, and you've muted the neurological drive required to snap it back. The muscle is long, but the brain's connection to it is muffled.
So if dynamic stretching, like leg swings or high knees, isn't enough to get someone into a full split,
Yeah.
and static stretching completely kills their explosive power, what on earth are these athletes supposed to do?
It's a huge dilemma.
I mean, think about a martial artist who needs to throw a lightning-fast head kick, or an Olympic weightlifter who needs to drop into the absolute deepest, most flexible squat position to catch a 400-pound barbell, but then needs maximum explosive power to stand back up.
Exactly.
That is the exact trap these researchers at Memorial University of Newfoundland wanted to break these athletes out of.
Yeah, and to find a way out of that trap, the researchers designed a highly specific, very rigorous experimental protocol to test a timing loophole.
Okay, let's hear about the setup.
They recruited 17 healthy, recreationally active young adults, specifically 9 women and 8 men, and they put them through this intense stretching intervention. When I say intense, I mean they really pushed the tissue to its functional limit.
Break down what that intervention actually looked and felt like for the participants.
Well, in the experimental condition, the participants performed static stretching on both their quadriceps and their hamstrings, and they didn't just touch their toes for a few seconds. They performed 4 separate repetitions of 45-second static stretches for each muscle group.
Whoa. So we are talking about 180 seconds. That's 3 full minutes of deep, continuous static stretching per muscle.
Yeah.
Anyone who has ever done targeted mobility work knows that 3 minutes is an absolute eternity when you are under tension.
It absolutely is, especially because the intensity instructions were very clear. The participants were told to stretch to the point of maximal discomfort.
Maximal discomfort. Ouch.
Right. I mean, they were not supposed to injure themselves, obviously, but they were taking the muscle exactly to the edge of its pain tolerance and holding it there.
Okay, so at the end of this 3-minute ordeal, They have effectively turned their legs into those overstretched floppy rubber bands.
Precisely.
They have numbed their muscle spindles. They have suppressed their neural amplifiers in the spinal cord. Under normal circumstances, if you asked them to do a vertical jump right then and there, their performance would completely tank.
Unquestionably. The literature shows they would experience a huge drop in power. But here is where the study introduces the loophole.
Okay.
After this intense, agonizing stretching session, the researchers just had them stop and wait for exactly 2 hours.
Wait, hold on. I have to push back on this premise just based on my own physical experience.
Sure.
They stretched until it hurt and then just sat around a laboratory for 2 hours.
Yeah.
If I sit still on a couch for 2 hours after a heavy workout or a deep stretching session, my body completely cools down.
Right.
My hamstrings turn into steel cables and I lock right back up. How does that make any sense? It seems completely counterintuitive. I would assume a 2-hour gap would do nothing but erase the flexibility they just worked so hard to achieve.
Your assumption is incredibly common. And it's actually the exact biological intuition the researchers were testing against.
Oh, really?
Yeah. The core hypothesis of this study was that the human body's physiological timeline for flexibility might operate on a completely different biological clock than its timeline for neuromuscular fatigue.
Meaning, the floppy weakness might wear off faster than the actual structural looseness. Like the hardware and the software are running on different timers.
Precisely. That is exactly it. So let's look at the data and address your assumption about the body tightening back up.
Okay, let's see.
The participants sat in the lab, read books, used their computers, and just relaxed for 2 hours. Following that rest, they did a standard 10-minute dynamic warm-up.
Just a standard warm-up.
Yeah, this was just 5 minutes of light pedaling on a stationary bike, some very brief 15-second maintenance stretches, and some dynamic movements like high knees. And then, finally, they were tested.
And what happened to the flexibility? Did it actually survive the 2-hour waiting period?
It did. The first major finding of the study was that the structural looseness remained.
Wow.
Compared to the control condition, which was a separate day where they just did the 10-minute dynamic warm-up without the 3 minutes of prior intense stretching, the experimental group retained a significant increase in their range of motion.
How significant?
Specifically, they saw an increase of about 4.1% in their right hip flexion and 4.9% in their left hip flexion.
That is fascinating. So the tissue compliance outlasted the rest period. But here's where it gets really interesting. What happened to the power? We know they kept the length, but did the 2-hour wait successfully reboot the nervous system and rescue that floppy rubber band effect?
It did more than just rescue it. It completely nullified the negative effect.
Seriously.
Yeah, the researchers rigorously tested the participants' power output. They tested their MVIC forces for both knee flexion and extension to measure absolute strength.
Okay.
They tested counter-movement jump height and drop jump height to measure explosive reactivity. And they used EMG, which is electromyography, to measure the actual electrical activation signals firing into the vastus lateralis in the quads and the biceps femoris in the hamstrings.
And what did the data show across all those power metrics?
Across the board, there were absolutely no significant impairments in any of those metrics.
None at all.
None. The maximal force output didn't drop. The jump heights remained identical to their baselines. Most importantly, the EMG electrical muscle activation was completely normal. The neural drive from the brain to the muscle was firing at 100% capacity.
So they actually did it.
Yeah.
By purposefully separating the deep, painful static stretching from the explosive event by exactly 2 hours, they allowed the body to compartmentalize its physiological responses.
Yes.
The nervous system recovered, but the musculoskeletal system stayed loose.
They discovered a highly practical way to basically hack the biological system.
So we've established the what. We know the 2-hour protocol works. But I really want to dig into the why and the how, because understanding the mechanism is how we figure out who this actually applies to.
Good point.
Let's look at the demographics of the participants. They specifically used 9 women and 8 men. Did this biological loophole work universally across the sexes? Because we know that baseline flexibility between men and women can be remarkably different.
That is an essential nuance, and it's something the researchers explicitly analyzed, particularly because female physiology is historically underrepresented in sports science literature.
Yeah, totally.
To understand the results, we have to clearly distinguish between absolute range of motion and relative range of motion.
Right, because if you just look at the raw data, you might get a skewed perspective.
Exactly. When looking at the absolute baseline numbers, the study found a large magnitude of difference. The women had significantly greater absolute range of motion than the men before the experiment even started.
Not surprising.
Right. The women were reaching around 122 degrees of hip flexion, whereas the men were maxing out around 100 degrees.
Which perfectly aligns with a vast body of existing literature regarding, hormonal differences, joint structure, and innate tissue compliance between the sexes. Women generally have a higher baseline of flexibility.
They do. However, the crucial finding of the experiment was that there was no sex-by-time interaction.
Meaning the protocol itself didn't favor one biological sex over the other in terms of how the body reacted to the 2-hour wait.
Precisely. The relative benefit of enduring the 180 seconds of intense stretching, waiting for 2 hours, and then doing a light warm-up was identical for both the men and the women.
That's great.
Yeah, they both achieved that roughly 4 to 5% increase in flexibility relative to their own unique baselines, and neither sex experienced a drop in explosive power.
So it is a universal human mechanism.
Yeah.
But let's get back to the physical reality of the tissue. How is the body actually managing to keep the physical flexibility but ditch the neuromuscular weakness?
That's the million-dollar question.
Right. Because if I stretch a rubber band and leave it on a table for 2 hours, it shrinks back to its original size. Why doesn't the human hamstring do the same thing?
Well, it comes down to a physiological race between different recovery systems.
Okay, a race.
Yeah. The negative effects of static stretching, that muscle spindle numbing, and the suppression of the neural amplifiers in the spinal cord, those are primarily neural and mental fatigue factors.
Okay.
And the human nervous system is incredibly resilient. It bounces back relatively quickly. Within that 2-hour window, the nervous system completely resets, the numbness wears off, and the brain-to-muscle connection is fully restored.
Okay, so the software reboots quickly. But why does the hardware, like the muscle tissue and the fascia, stay elongated?
Because the physical compliance of the tissue simply takes longer to revert. The actual decreased stiffness of the muscle fibers and the tendon structures persists on a longer timeline than the neural fatigue.
Oh, I see.
Additionally, there is a major psychological component at play.
A psychological component. How so?
Your mental tolerance to the stretch, your perception of where the pain point begins, has been pushed further out. That psychological tolerance sticks around longer than the neural suppression.
The hardware stays loose, the software reboots, and your brain's fear of the stretch has been quieted down.
Exactly.
That is an incredibly elegant biological solution. But I imagine the dynamic warm-up they did right before the testing played a role in this too, right? Like they didn't just stand up from the couch and immediately try to do a max effort vertical jump.
You are absolutely right to point that out. The traditional dynamic warm-up they performed just prior to testing the 5 minutes on the stationary bike, and the dynamic leg swings that likely provided a crucial rescue effect.
Let's explore that rescue effect. What is 5 minutes on a stationary bike actually doing to the tissue that was stretched 2 hours ago?
Well, by physically moving the limbs and pumping blood into the area, you are increasing the core tissue temperature. And this induces what biomechanists call thixotropic effects.
Thixotropic effects.
Yep.
Okay, think of it like stirring a can of thick paint or squeezing honey out of a bottle.
Perfect analogy.
When it's cold and still, it is highly viscous and resistant to movement. But the more you agitate it, move it, and warm it up, the more fluid and less viscous it becomes.
That is the exact principle happening inside your muscle fascia. The physical movement reduces the tissue's resistance to movement. Furthermore, the dynamic warm-up actively excites the corticospinal pathways,
Which is the main communication highway traveling from your motor cortex in your brain, down your spinal cord, and out to your muscles.
Exactly. By firing up that highway with dynamic movement, any tiny microscopic bit of lingering neuromuscular slowness from the deep stretching 2 hours prior is completely overwritten by the new, highly active neurological signals.
Its entire protocol is incredibly actionable information. If you are listening to this right now and you participate in a sport, maybe you have a martial arts class tonight, or a gymnastics meet, or an Olympic weightlifting session where you absolutely need to drop into a deep, flexible position, but you cannot afford to lose your explosive power.
Mm-hmm.
This is your new blueprint.
It really is.
You don't have to choose between flexibility and power anymore. You just have to manage your biological clocks. You separate your deep, painful static stretching from your actual event by a minimum of 2 hours.
It completely reframes how athletes and coaches should schedule their event preparation.
Total.
Instead of doing your intense, agonizing stretching on the mat right before the competition begins, which we now know actively harms your power. You do it in your hotel room or at your house 2 hours prior. Then you commute, you rest, you do your normal light dynamic warm-up at the venue, and you step into your event with the best of both worlds.
But applying this blueprint leaves me with a massive provocative question about the boundaries of human physiology.
Oh, let's hear it.
If the neural inhibition fades in 2 hours, but the structural looseness remains, what is the absolute biological limit of this effect?
That is the perfect question to ponder as we look toward the future of sports science.
Right, because if the software reboots in 2 hours, but the hardware stays loose,
Yeah.
how long does the hardware actually maintain that compliance?
Could an athlete do their deep static stretching 4 hours prior? What about 6 hours?
Exactly. And if they take a nap in between, does sleep reset the tissue tension faster?
We just don't know yet. We know that 2 hours is a proven biological sweet spot, but we don't yet know where the cliff is before the tissue inevitably tightens back up to baseline.
It makes you wonder how many records could be broken just by tweaking the timing of a warm-up by an extra hour. But until researchers test those extended time frames, the 2-hour loophole remains a scientifically proven tool that you can add to your arsenal today.
Absolutely.
We highly encourage you to read the full article for yourself to truly grasp the depth of this research. Again, the study is titled The Effects of Static Stretching 2 Hours Prior to a Traditional Warm-Up on Performance by Shea, Bahrami, Sardroodian, and Behm, published in the Journal of Sports Science and Medicine in 2024.
Yeah, check it out to see all the precise data charts, the exact electromyography readings, and the full details of the testing protocols.
It is an accessible, rigorously designed study that challenges some of the most deeply held assumptions in our field. And it proves that sometimes timing is literally everything.
It really is.
Make sure to follow the channel and turn on your notifications so you never miss out when we explore these kinds of actionable scientific breakthroughs. Thank you for learning with us today. Remember, the human body might seem like a paradox of conflicting needs, but sometimes, if you look closely enough at the science, you can find the loophole